So welcome everyone to the Science on Sundays talk for today. My name is Neha Bhatia. I am a group leader at the Sainsbury Laboratory just next door, just building to this beautiful garden. And today I'll try to share with you what I have learned through research about patterns in form that we see around us in nature. So I'll very briefly brief you about my academic journey so far. I come from India, where I studied my bachelor's and master's in plant sciences. Then for my PhD, I moved to Germany at an institute called EMBL in Heidelberg. And then after completing my PhD in 2016, I briefly moved to Australia, where my PhD supervisor's laboratory had shifted to continue finishing some projects that I had started in my PhD. And then after spending 18 months there, I moved back to Germany again to start my postdoctoral studies at an institute called Max Planck Institute for Plant Breeding Research in Cologne. And then since November 2024, I've started my own research group at the Sainsbury Laboratory. I am a developmental biologist by training, which means that I like to study fundamental sciences and how organs form and grow in plants. So although development in plants and animals is a very different phenomenon because we belong to different kingdoms, yet there are some common principles that underlie formation of organs like leaves from plants and fingers in a human hand. And these are a system called positioning, which ensures that leaves originate at correct positions around the plant stem. Like our fingers, they initiate at correct positions. They are not forming from anywhere, but in this special position. Then they have to become specialized so then they can execute their function. For example, leaves have to acquire certain structures so that they can do their function like photosynthesis. And then fingers also have to acquire, they have to develop bones so that they can hold things. They have to develop muscles so that they can function properly. And then finally, they have to grow to a correct size. That means that we don't have out of proportion hands or fingers. And in nature, we see that even in this picture here, that you see that all the leaves are almost of like similar size. They're not going or competing in a very different way from each other. And I have been very lucky throughout my research career to have touched upon on all these three core principles of how organs develop. But today I will be mainly talking about a system called organ positioning. Now I'm sure if you take a walk around this beautiful garden, you may have noticed that there are diverse plant forms around us, and they show very striking arrangement of flowers or leaves around the stem. And it was already studied in 1990s that the spiral patterns of organ development and positioning are most common. So leaves or flowers can orient perpendicularly to each other, like opposite, or they can go and form these very intricate and beautiful spirals. And this process is called phyllotaxis, which means the symmetric arrangement of plant organs around the stems. Phyllo means leaves and taxis means arrangement. It's derived from a Greek word. Now, as you can see that these arrangements are so symmetric and precise that it has captivated interests of not only biologists like me, but also mathematicians, physicists, and even computational scientists. And so there have been attempts made to explain how spiral organ formation may originate. So here is a physical demonstration of spiral phyllotaxis. So what people did here, that they took a petri dish, like a glass dish, and they put silicone oil in that, and they created magnetic field around this glass dish, and they started to drop iron-containing droplets in this field. Now, if you see that these droplets at the moment are dropped very slowly, and all they are doing is just moving away from each other without forming any pattern, just drifting apart. If when they increased the frequency or the speed with which droplets were dropped, they could organize into these spiral patterns, which became more obvious when the speed with which the droplets were dropped was increased further. So this suggested that the frequency in which droplets were formed helped them organize into certain patterns, and this could be translated into plant organ formation, which means that regular spacing arising as a result of the frequency with which they are formed could create organs in a regular manner. Now, this is all like physics, and I am a biologist, so I want to understand the biology behind how these patterns emerge. So, I cannot use these beautiful cacti in a laboratory because they are very big, and then it is hard to grow them and do research within a limited time frame that we have. So we make use of a model organism called Arabidopsis thaliana. It's a common weed which you would see growing around even in the botanical garden or in your garden at your home. So it's a very simple plant. It has a life cycle of seed to seed, roughly 2 months. And we know many genes in this plant and many mutants, which means that plants in which function of certain genes and proteins is not functional have been characterized and they are available. So this helps us study this plant easily. Now, if you look at the growing tip here, this is where the organs like flowers initiate. And if you take a look at it in a close-up, you see that all these flower buds are arranged in a certain manner. Now, I can see that you do not understand how this is a spiral here, but I will come to that in a few slides, so please bear with me. Now, these flowers originate from a structure called shoot meristem, which you see in the green here, and these flowers are initiating at the flanks of this structure here. So if we remove all these mature flower buds, we can access this structure which gives rise to these flower buds, which you see here as very transparent thin forms. These are like babies of flowers, let's say, in their initiating form. And this is the structure, dark green colored, which is called the shoot apex or the shoot apical meristem, or SAM, as I will call it in my slides from now. And it's the same for leaves. Leaves also continue to originate from this shoot meristem. So you can see that we can grow lots of Arabidopsis plants as seedlings in the lab. And just to give you a reference, these are really tiny, even after 11 days old. Compared to my fingernail here, we can have many, many growing in a petri dish. And if we take a closer look, this is less than 1 millimeter. It has already formed 2 embryonic leaves and then leaf 1 and 2. And if we remove these leaves, we can see that it has formed, it is continuously forming new leaves, which is like, so I remove leaf 1 and 2, and then I remove leaf 3 and 4, and these are leaf 5 and 6. So the beauty of studying plants is that most of their organs form post-embryonically. I'll just wait a minute. Yeah. So most of these organs form post-embryonically, means after they have passed their embryonic development. Unlike animals or humans like us, we are born with our hands, our feet, and all organs already developed. But plants continue to make new organs throughout their life cycle, and this allows us to study how organs form and emerge and develop from the very beginning. Okay. So how can we understand the process of organ initiation like flowers and leaves from the shoot apical meristem? One way is to identify plants that do not form organs in this regular fashion, as in what we call genetic mutants. One such mutant plant that you see here is called PIN1. And the reason it is called PIN1 because Here you have a control plant, which is very normal. It is forming flowers. But in this plant, you see that the shoot is continuously growing as a pin without any flowers. And so that's where the name pin1 was originated. And this mutant, in fact, was already documented in 1959 by Capert. You see that it has no flowers and the stem is just growing like a pin. So what is wrong with this mutant? Here you see a very high resolution microscopic image of the plant growing tip. This is the shoot meristem, and these are young flower buds. And in this mutant, you can see that the shoot meristem, as I showed in the big plants, there are no flower buds in the vicinity. But when scientists used plant hormone called auxin, and they mixed it with a paste similar to Vaseline, and then they just applied a drop of that paste containing auxin onto this shoot meristem, they could restore flower formation. So you see, now flower is originating from the shoot meristem, and this is the paste, which means at the site where auxin was applied very locally, flower formation was restored. So this experiment led to a very important discovery that local auxin accumulation at the SAM is required for local or regular organ formation. Now, how would that happen? So, because pin protein does not form flowers and it formed flowers upon local auxin accumulation, it suggests that this plant has a problem in concentrating auxin locally, which means that this protein called pin1, which is not functioning in this mutant, might be playing a role. So we wanted to visualize what is this pin protein and what exactly it is doing in the plants to help mediate this auxin accumulation. So how can we do that in living plants? You may have learned or known jellyfish, which glows in dark. So it can do that because of the presence of a special protein called green fluorescent protein in it. So what scientists did, that they could extract this green fluorescent protein and sequence it to have the information how this protein is made. So in the lab, what we can do, that we can take whatever protein that we want to study, along with its managing elements that help the protein to function, and then we can fuse it to, it's like you are making cookies. So for example, if you want to make a green cookie, you can add a green dye to it, so it will become green. If you want to make like different colors, then you can add whatever dye you want to it, and then you can get similar colored cookies. So we can do same thing with proteins. We can fuse them to this green fluorescent protein, and it creates this fusion protein that glows when we shine special laser onto it. And this allows us to visualize multiple proteins at the same time in plants. For example, this gene A or protein A in pink is localized here, and this protein G called B, which is shown in green, is like outlining individual units of this leaf called cells. So using this technique, we were able to visualize this protein called pin1, and what we found that it localizes on the cell outlines. We call them as membranes, but you can imagine them as skin of cells. For example, if you blow air in a balloon, whatever is on the outer surface, it's holding the air inside to give it a shape. And so these cell membranes are essentially found also in our cells, and then they just help plant cells or animal cells to hold their shape. And if I play this video, you will see that over a time frame of 12 hours, this spin protein is changing size. So it is initially localized, if I play this video again, on this side of the cell, and later on it has, you see more signal on the other side of the cell. And maybe this is more clear in this snapshot from this video. This is the first time point when we saw it. You see a lot of orange here on this side of the cell, and at a later time point, orange has disappeared from here, and most of the orange is now seen on this side of the cell. So it has flipped its side or localization. Now, this protein can do this changes in sites in a collective manner. So in a way, cells can change the orientation of their pin proteins together in a collective manner. So if you focus on this video here, all these cells are changing the pin 1 or pointing this pin 1 towards this side like that. And then cells below this dash line are sort of trying to form this kind of a pattern, like converging from all different directions over here. And this indicates that plant cells can talk to each other, like each of these cells was coordinating that together we have to orient or change the side of our pin protein to one particular side. Now, what is this pin protein and why is this important? So this pin protein is actually a transport protein. It's like a pump, which when localized on this side of the membrane, it transports the plant hormone auxin outside of the cells into the neighboring cells. So, and the direction of its localization or the sidedness of its localization determines the direction in which auxin is being pumped or exported out of the cell. So you see that this is one single cell here, and it is localized halfway through the cell, the orange signal. You don't see it all around the cell, but only halfway through the cell in the form of these arc. And by doing this, this protein is transporting the hormone auxin outside from this cell into all the neighboring cells that will be here. So, with all this information in mind, what you're looking at here is the shoot meristem from a very high, this image has been taken with a very high resolution microscope where we can visualize the localization of pin proteins in each of these cells. This is a very complex image, but let me walk you through what you're looking at. First thing is that you see that I have used an intensity-based gradient color, which means wherever there is more hot color, like very yellowish or white, there is a lot of protein here. And when the color is very low, like purple or blue, then there is low protein here. So that means at certain positions in this shoot apex, there is lot of pin-1 protein, and then at certain positions, there is very little pin-1 protein. And if we go back to these arcs, this protein sort of forms by localizing at certain sites these guided patterns. Like you see when you look at the air traffic control from satellites, you see the direction of all the planes moving towards different direction. Cells are also able to do that by shifting their proteins in these intricate directions. So you see that some of these cells like forming cell arrows, and all these arrows are pointing towards this site at this shoot meristem, and some arrows are pointing towards this site at the shoot meristem. Some are pointing like here, but some are pointing away from here. And so what exactly is happening with the help of these guided arrows? Local accumulation of this plant hormone auxin, because this protein transports auxin outside from the cells into the neighboring cells. So by forming these guided patterns or transport routes, it creates auxin hotspots at different positions in the shoot meristem. And this is where organs emerge at these sites. So new leaves and flowers will form from these sites from where auxin hotspot has been created. And the beauty of studying plants in that sense is that you can look at organs at different stages of their initiation within the same image. So you can see that organ in position 1, organ at position 2, 3, 4, 5, 6, 7, and so on. So you see at least 7 different organ positions within this same image. And coming back to the spiral, so if I just draw lines from position 1, 2, 3, 4, 5, 6, 7, it's a spiral. So that means that plants are creating these organs in a spiral manner at regular positions. And so this is a very simplified version of everything that I showed, that auxin shown here in form of these green dots is accumulated at certain sites, which is guided by the transport patterns of this or transport routes of this hormone. And then from here, these organs emerge like flowers. So the question that I wanted to ask, or that was very interesting to me, was how do plant cells decide where do they have to guide auxin transport? So how do they channel auxin transport to certain sites to create auxin hotspots from where new flowers or leaves will form? Now, as I mentioned, that this problem of plant organ formation has not only attracted biologists, but also computer scientists. So computer scientists came up with a theory that a cell eye can, like you just see a group of cells, it's a very hypothetical or like a theoretical part, not actual cells here. There are two cells, cell I, and it has 4 neighbors. So among all of its 4 neighbors, this cell I can somehow sense that the neighboring cell J has most auxin or highest amount of auxin compared to all my neighbors. And so I decide to orient most of my PIN1 or the transport protein towards this side of the cell, which is next to cell J. And this essentially creates a positive feedback loop where auxin controls itself where the transport direction will be guided. So more auxin here will help this cell I to transport more auxin further into this cell, creating this positive feedback loop. And it is the one way to test this theory would be that you increase the amount of auxin in one cell and then see whether the neighboring cell can change its PIN protein localization towards the site of the cell which has high auxin. This is very difficult to do because plant hormones, like other hormones, are very mobile. So even though they require special proteins through which they can be actively transported, they can also diffuse freely from the cell. So there is no guarantee that if I increase auxin in one cell, it will stay there for very long. So we have to find solutions. So what we can do instead is, like, we can make use of the messenger proteins through which this hormone might be acting. And those messenger proteins are called signaling proteins in our day-to-day laboratory language. So when there is no auxin, these messenger or signaling proteins are present there in the cell, but they are sleeping. But when auxin is present, they suddenly become very happy and are very active and are able to mediate the function of the plant hormone auxin itself. So we created a hypothesis or like a idea that how this feedback loop between auxin and its transport protein could be functioning. It could be that auxin would then tell its messenger protein that go and change the direction of the PIN1 protein, and which will then mediate this feedback onto this hormone auxin itself. So does that happen? It can happen, and there are some messenger proteins which were already known by the time when I was studying my PhD or doing my PhD. So I told you that this PIN protein, when is not functioning in the plant does not form flowers. But when we apply auxin very locally, it forms flowers. But in this plant where the signaling protein is not working, it does not respond to auxin. So even upon providing this plant with auxin very locally, it does not initiate flowers. So that makes sense because that means that auxin messenger protein is required to mediate local accumulation of auxin itself. And that's why there is no organ forming here. We can actually test that how cells decide, or these proteins decide, which side of the cell we should go. So what experiment I did was, I activated this auxin signaling protein in only one or a few cells, and then I could see whether the neighboring cells can then reorient their PIN1 protein, which is now maybe on all sides of the cell, to the side where this auxin signaling is active. So this is like a proxy of high auxin concentration itself. Because there is auxin signaling protein active, which means that there is auxin in this plant cell. And indeed, this did happen. So what you see here is the cell where I induced or I increased auxin signaling, which you see here in green. So before I could see, I did this experiment, I could see that the neighboring cells, most of their PIN protein, like you see all of the purple or magenta on this lateral side of the cell. After 6 days of the experiment, when it was conducted, when I increased auxin signaling very locally in this cell, which you can see in green here, most of the signal from the side has shifted to the side next to these green cells. And in this process, you can see that these two cells have become 4 cells. So this was done in living plants, and plant cells continue to divide over time. And so all these mother cell and their daughter cells could change their transport protein localization from the sides to the side of the neighboring cell where there was high auxin signaling activity or high auxin activity. So your take-home message 1 from this work is that if a cell detects a lot of auxin, it makes the neighboring cells transport auxin towards that cell. And this creates an auxin hotspot from where new organ can emerge. Now, things are not as easy when it comes to forming organs because nature is very complex. So you see that there are auxin hotspots created here, and some arrows are like in the arrows of the transport are forming this kind of a pattern to concentrate auxin. But then one more thing before that, what you see in the inside of this dotted black outline is the center of the shoot meristem. These 3 organs here are very young, so they have just been positioned, whereas these organs have already grown out. So they are emerging out from this center of the shoot meristem. You see that they have bulged out from this central structure. And if you notice that they have auxin at their growing tips, which helps them to grow out. But then on the other side, these arrows are sort of pointing away from this organ. That means that once auxin has been accumulated and a hotspot has been created, it just cannot stay there forever. It has to be transported to different sites away from this. And so once this organ has grown out, the machinery activates and functions in such a way that it transports auxin away from this site to the new or the next organ formation site. And this process does not only happens on the surface, but also below the surface. And when I say below the surface, I mean like when you buy a fondant cake or you bake a fondant cake, you see that it has, if you slice it, you will see that it has layers in it. It's the same for plants structure. They are three-dimensional structure. So if I make a slice or a section through this emerging flower bud, what I will see that there are channels in the deeper tissues where auxin has to be drained away from the surface. So once auxin is accumulated and the organ has grown out a little, auxin drains away to the next organ initiating site, but also it has to go deeper into the tissues. And so why is this important, and what happens when auxin just stays there and does not go deeper inside the tissues or away from the site? So this is the normal Arabidopsis shoot that has given rise to separated flowers here. And when auxin drainage is not happening, you see that it gave rise to organs that were running like a spiral band all around the tissue with no separated flowers forming. So like that, like that, like that, like that. And this looks very similar to some of the twisted serious cacti that we see in the nature, like this. So they are forming spirals, but then they are not forming it in correct manner, and organs grow continuously conjoined with each other as a continuous band, as opposed to these discrete flowers. So we wanted to then see that what is wrong with auxin in these plants, that what is exactly happening. If it is not going deeper inside the tissue, then it has to do something in the plant. Now, visualizing hormone is not as easy. We could have visualized the signaling proteins as well, but then this would not really be helpful. So what we did was, we took help of computational scientists, where we provided them all the information that how we have created this plant, and then they could impart that information to a computer model and then show us what exactly was going on. And through this, what we found, that instead of going deeper down, the two auxin peaks you see here in green were just continuously moving around, round and round and round, and leaving behind trails of that organ behind as they were moving. So just spiraling down the stem, and then as they were doing that, they were leaving behind these trails of continuous organ bands, and that gave rise to this spiral. So the take-home message too is that after auxin hotspot has formed, auxin levels need to deplete in the surrounding cells, which you see here, from this organ into this, and to contribute to forming of the auxin hotspot at this new organ, and then also deeper inside. And this allows the plant to form a new auxin hotspot at a distance from already formed flower. And this creates organs in a spiral fashion, and which are regularly spaced from each other. And you'll be very interested to know that this mechanism not only creates organs like flowers and leaves from the SAM regularly, but it also helps to create these leaves that you see in your salad, like rucola, where we can have subgrowths growing or outgrowths growing from the margin of the leaves that have initiated. Okay. So what I would like to convey finally is that studying fundamental science is very important, because it helps us understand the diverse forms that we see around us and appreciate what nature has created for us in a very complex manner. So with this, I would like to thank you for your attention. I did not show you what I am trying to investigate or seeking in my new lab here, but maybe in the coming years when I have developed something substantial. Meanwhile, I would like to thank all the people, my supervisors with whom I have worked with. Miltos was my postdoc supervisor based in Cologne, and Marcus was my PhD supervisor, who is now in Australia. And then I would like to thank SLCU for giving me this opportunity to establish my own research program. And thank you for your attention. I'm happy to take any questions that you may have.